In this video, we will learn about the structure and the function of the nucleic acids, DNA and RNA. We'll learn about their similarities and their differences, and we'll learn about their molecular subunit, the nucleotide. Nucleic acids are one of 4 types of biological macromolecules. Nucleic acids are specifically adapted to store and transfer information. Nucleic acids got their name because they were initially found in the nucleus of the cell. There are 2 types of nucleic acids, DNA and RNA. Even though they were initially discovered in the nucleus of eukaryotic cells, nucleic acids exist in all living things, including prokaryotes, which don't possess a nucleus at all. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. These 2 molecules are closely related, but they have several important differences in their structure. This makes them adapted to related but different functions. Let's begin by examining the general structure of a nucleic acid. Nucleic acids are polymers, which means that they are large molecules that are made up of several repeating molecular subunits, or monomers. The monomers of nucleic acids are called nucleotides. The nucleotide itself is made up of 3 parts: a phosphate group, a pentose sugar, and a nitrogen-containing base. The phosphate group is always the same. There are 2 types of pentose sugars found in nucleic acids: ribose sugar and deoxyribose sugar, which contains 1 less oxygen atom than ribose sugar does. As you can likely tell by their names, deoxyribose sugar is found in the nucleotides of DNA, and ribose sugar is found in the nucleotides of RNA. Finally, there are 5 types of nitrogen-containing bases. They are adenine, guanine, cytosine, thymine, and uracil. The nitrogen-containing bases are often represented by their initials. The base thymine is only found in DNA, and the base uracil is only found in RNA. Adenine, guanine, and cytosine are found in both. The polymerization of nucleotides is what makes a nucleic acid. The pentose sugar of one nucleotide bonds with the phosphate group of the adjacent nucleotide. The atoms of their 2 hydroxyl groups rearrange so that both molecules are sharing 1 oxygen atom. The 2 hydrogen and 1 oxygen atoms that are released in the process bond together to form 1 molecule of water. This chemical process is called a condensation reaction. The resulting bond between the phosphate group and these 2 sugars is called a phosphodiester bond. The bonds between the pentose sugars and the phosphate groups form a stable structural chain, which is referred to as a sugar-phosphate backbone. Well, we know that nucleic acids are adapted to store and transfer information, and that information is contained in the sequence of the nucleotide bases. And in order for that information to make sense, that sequence must follow a specific order, which brings us to a concept called directionality. It's common scientific convention to number the carbon atoms in a sugar molecule. The carbon atoms in our pentose sugar are numbered 1 through 5, like this. The 5th carbon is bonded to the phosphate group of its own nucleotide. So this end of the molecule is called the 5' end. The 3rd carbon atom in the pentose sugar is bonded to the phosphate group of the adjacent nucleotide. So this end of the molecule is called the 3' end. We read nucleotide sequences from the 5' to the 3' direction because that's how the polymerization occurs in living organisms. Let's practice quickly by writing down the nucleotide sequence from our example. From 5' to 3' it reads G, A, T, C, G. Now that we've learned about the general structure of nucleotides and how they're able to form a sequence of readable nucleotide bases, let's look at the specific structure of DNA and RNA. DNA is a nucleic acid that's specifically adapted to store information and to pass that information on to offspring cells or organisms. DNA is made of 2 strands of nucleotides bonded to each other at their nitrogen-containing bases. Because of the structure of the molecular subunits, DNA forms a twisted ladder shape, also called a double helix. The sugar phosphate backbone makes up the sides of the ladder, and the bonded nitrogenous bases are the rungs. If we untwist this ladder, we can see the nucleotide sequence more clearly, and I've also added the letters that represent the nitrogen-containing bases. If we zoom in a little more closely on this structure, we can see that the molecules in one strand are facing in one direction, and the molecules in the opposite strand are pointing the opposite way. For this reason, one strand reads from 5' to 3'. While if we read in the same direction on the opposite strand, we're going from 3' to 5'. This arrangement is known as anti-parallel, since the 2 DNA strands are aligned but oriented in opposite directions. The direction that we read a DNA strand in is important, since the sequence of the nucleotide bases is what determines the genetic information. Another feature of the DNA molecule is that each of the nitrogen-containing bases only bonds with a complementary match. In DNA, adenine always pairs with thymine, and cytosine always pairs with guanine. You'll also notice that a larger base always pairs with a smaller base. This allows the DNA molecule to maintain the same width along its length. This is what we refer to as the base pairing rules. I've left some blanks on our example DNA strand so that we can practice base pairing together. If the nucleotide sequence on one strand of DNA, reading from the 5' to the 3' direction, is C, T, A, then what would be the sequence on the complementary strand, reading from the 3' to the 5' direction? Well, the complementary nucleotide sequence would be G, A, T. The complementary strands of DNA are held together by hydrogen bonds. Unlike the phosphodiester bonds found in the sugar phosphate backbone, these hydrogen bonds are relatively easy to break. All of these features of DNA are what allow it to be replicated quickly and precisely, which is essential in its function as hereditary information. The function of RNA differs from that in DNA because RNA is adapted to transfer information from place to place. We're also already aware that the RNA nucleotide includes ribose sugar instead of deoxyribose sugar. mRNA is responsible for carrying information from the DNA where it's stored to the ribosome, which translates it into a protein. In order to accomplish this, mRNA follows a similar set of base pairing rules to DNA. Here, we have a molecule of DNA where the 2 complementary nucleotide strands have been separated. In order to transfer the information, a complementary RNA strand must be built. Just like in DNA, small cytosine always pairs with large guanine. And where we have thymine on the DNA molecule, adenine will always be the complementary RNA base. But where DNA possesses adenine, the complementary RNA base will be uracil instead of thymine. Why don't you try to fill in the remaining complementary RNA bases on your own? Once the RNA has copied the information from the strand of DNA, it detaches and travels to the ribosome, where the protein synthesis process begins, and the 2 DNA strands rejoin. Besides the differences that we've already noted, we can also see here that DNA tends to exist as two bonded strands of nucleotides, while RNA tends to exist as a single strand of nucleotides. Also, in eukaryotic cells, DNA is stored in the nucleus, while RNA can be found inside of the nucleus and in the cytoplasm. Now that we've learned about how the differences in the structure of DNA and RNA support their different functions, let's try a practice question. DNA and RNA are both examples of nucleic acids. Compare and contrast the structures of DNA and RNA. This question is asking us to compare, which is to describe similarities, and to contrast, which is to describe differences. And they want us to describe the similarities and the differences between the structures of DNA and RNA. And we're being asked to specifically focus on the structure or the parts, and not the function or what it is these molecules do. One way to organize your information when you're working on a compare and contrast question is to make a table with a column for DNA, a column for RNA, and a column for both. Then you simply fill in whatever you know, which will help you to write out your answer. Another way to organize your information is to make a Venn diagram with a circle for DNA, a circle for RNA, and information about both where the two circles overlap. I like a more visual approach, so I'm going to continue with the Venn diagram, but you can choose to use whichever method is most comfortable for you. I've also added some visual clues, which we'll annotate together before writing out our final answer. Let's start by noting that this question already told us that DNA and RNA are both nucleic acids. And we know that nucleic acids are polymers, and that a polymer is a large molecule made up of repeating subunits, which we call monomers. The monomers of nucleic acids are called nucleotides. And each nucleotide is made up of a phosphate group, a pentose sugar, and a nitrogen-containing base. However, DNA is made of two strands of nucleic acids, bonded together into a double helix, while RNA is typically a single-stranded molecule. There are also some differences between the nucleotides of DNA and RNA. The pentose sugar in the nucleotides of DNA is deoxyribose sugar, while in RNA it's ribose sugar. Also, the bases in DNA are adenine, thymine, cytosine, and guanine. RNA molecules possess adenine, guanine, and cytosine, but the base thymine is replaced by the base uracil. Now that we've summarized the similarities and differences in the structure of DNA and RNA in our Venn diagram, we're ready to write out our answer. Your answer, of course, will be written in your own words, but it should look something like this. Both DNA and RNA are polymers formed from multiple subunits called nucleotides. DNA and RNA nucleotides are both formed from a pentose sugar, a phosphate group, and a nitrogenous base. However, the pentose sugar in DNA is deoxyribose, and in RNA it is ribose. The nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine, but in RNA, thymine is replaced by uracil. DNA also forms a double-stranded molecule, whereas RNA forms single-stranded molecules. Let's wrap up our lesson by taking a moment to review what we've learned. In this video, we learned about nucleic acids, which are polymers made up of nucleotide monomers. We learned about deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. We learned about the structure of both. We also learned how those structures are related to their functions. The function of DNA is storage of information, and the function of RNA is transfer of information.